So you may be wondering, what are antigens? And antigen stands for antibody generators. That's where I got the name. And antigens are usually big complex molecules, and they usually have more than one site that can bind an antibody. But an antigen can also be very small if it combines with another carrier, such as a protein. And we call this small molecule a hapten. And for example, penicillin allergy is from penicillin binding to a protein in bacteria, and then the body making antibodies to the protein, and then penicillin. And I'll give you an example of this in a second with benzene. And it's also possible to have antibodies that bind polysaccharides or glycolipids. So I want to show you how the immune system usually works. So you usually have a protein, and then you inject it in a mouse, and then the mouse will make antibodies to the protein. In contrast, if we injected a small molecule like benzene and you inject it into the mouse, it is not going to make antibodies to benzene. But if we were to conjugate or attach benzene to a protein, and then we injected this benzene protein complex into the mouse would make antibodies to both benzene and to the protein. And that's a way of getting an antibody that will recognize benzene by conjugating it onto a protein. And this plays a really important role in immunity, because sometimes we have other surface structures other than proteins that we want the immune system to be attacking to give immunity. Maybe, for example, like polysaccharides on the surface of a bacteria. If you conjugate it to a protein, then the polysaccharides, then the body can make antibodies to those polysaccharides. And it does play an important role in immunity for some human diseases, such as those caused by pneumococci, for example. So I just want to remind you that the non-specific phagocytosis can be facilitated by macrophages, right? They just recognize common surface structures, nothing too specific. However, B cells can only phagocytose something with that exact 9 amino acid epitope. And if a bacteria has antibodies or complement bound to it, there'll be more efficient phagocytosis. And I like to think of macrophages being a little bit formal, kind of like the British, right? Like they like to eat with a fork and spoon. So I like to think of an antibody as a fork and complement as a spoon that facilitate the phagocytosis process for macrophages. And again, the important thing, whether there's opsonization or just regular phagocytosis, we have the microorganism entering the phagosome, it fuses with the lysosome, and then it gets destroyed. And then this 9 amino acid peptides can be presented on an MHC2 to facilitate antibody production. And I also just remind you that any B cell and T cell that have the ability to bind an exact 9 amino acid epitope found in our bodies, those cells are killed by an apoptotic mechanism. There's a special mechanism in our body that triggers apoptosis of B cells and T cells that have the ability to bind an antigen, a 9 amino acid epitope that is an exact match for the ones found in our bodies. The only B cells and T cells that can make it through the differentiation process and maturation process are ones that don't have an exact match. So I showed you the example with OspA. There was a 5 out of 9 match because it wasn't a perfect match. It was a partial match. Those can make it through and be B cells and T cells in the body that are capable of leading to the B cell producing a big antibody response. So I just want to go through this another time because it's really complicated, okay? So we know macrophages have both MHC1 and MHC2 on their surface. If a peptide is presented on MHC1, we know that that protein is from the cytoplasm of the macrophage. There will usually be a macrophage or human protein if the macrophage is not infected. If it has an intracellular parasite, then it could be a foreign peptide like a virus or bacteria. if the MHC2 protein has another peptide on the surface, we know that came from the phagolysosome, and that is going to be a foreign peptide only. So what we really see in this picture is it's a great figure to test your understanding, because it has the two scenarios here. So if the T cell here is a CD4 helper T cell, that's the CD4 receptor, we know that this is going to be an MHC2, meaning the peptide came from the phagolysosome in the macrophage. Okay? If this were a CD8 cytotoxic killer T cell, this is going to be a CD8 receptor, and then this is going to be an MHC1. And I just want to remind you that the T cell receptor can either see MHC1 or MHC2. It can't interact with both of them. When the T cell, be it a CD4 or CD8, sees its 9 amino acid peptide that it evolved to detect, then they will proliferate. They will replicate to be not one cell will become thousands of cells, right? And the cytotoxic killer T cell will look for more cells in the body that have this parasite in the cytoplasm of human cells, right? And it's going to go in to kill them using perforin and granzyme. And the CD4 helper T cell, in contrast, is going to look for its partner B cell that has hopefully already phagocytosed its microorganism that it can detect, and then presented the peptide on the MHC2, so that can create the plasma B cell and memory B cell and memory T cell. And here we go through this one more time, right? Because it's just so important. I know it's complicated, but it's so important that you all understand this. And here is a cell, okay? So we see the two pathways, the helper T cell pathway and the cytotoxic T cell pathway. So let's do the cytotoxic pathway first. So here's a macrophage. If the peptide is being presented on a MHC1, we know that that came from the cytoplasm, because a cytotoxic killer T cell is seeing that peptide. And this is suggesting that this macrophage has been infected by a microorganism that is now in the cytoplasm of the macrophage and is compromised the macrophage. So the cytotoxic killer T cell that has the ability to see this one 9 amino acid peptide, and to be activated, it'll end up secreting the perforin and granzyme that'll kill the virally infected macrophage. It'll also proliferate to make thousands of these cytotoxic killer T cell so that it can go and kill other virally infected cells. Now, in contrast, if this is a helper T cell, this is the helper CD4 helper T cell is going to see antigens in MHC2. That means the peptide came from the phagolysosome fusion, and then when the T cell finds this exact 9 amino acid peptide, it will proliferate, right, making thousands of the same T cell. It'll travel through the body, likely in the lymph nodes or spleen. It'll find its partner B cell that has already phagocytosed the entire microorganism. And critically, it can only phagocytose the microorganism if the antigen is on its surface. And that's why the B cell specifically helps our body make this amazing immune response to surface structures. Immunology is all about surface structures. I can't stress that enough. If it's not at the surface, the immune system can't see it. The immune system can only see things on the surface. So please think about surface structures when you're thinking about immunology. That's like just such an important concept in this field. So anyway, the B cell can phagocytose the microorganism attached to the antigen of interest, presented on the MHC2, and then the helper CD4 T cell will recognize it. They'll secrete a cytokine storm. The cytokine storm will basically lead to the plasma B cell, which I call the pregnant B cell, secrete thousands of antibodies per second. And then a memory B cell and memory T cell will be formed that can give long-term immunity. So I want to jump back to something I talked about in the beginning, which was the LPS endotoxin acting as a super antigen. So I want to introduce the superantigens are, and what I've been stressing in the last few minutes in this video have been the intricate interaction of a MHC2 with a helper T cell receptor, right? And how the T cell receptor will only activate and secrete cytokines when there's a 9 amino acid epitope being presented, a very specific one and only one 9 amino acid epitope presented on the MHC2. What a superantigen does is it ruins the specificity of that interaction. So it doesn't matter if this T cell can bind this peptide or not, right? It doesn't matter. The superantigen can facilitate vasodilators to be secreted by the T cell and macrophage. It doesn't matter if there's no peptide there or a foreign peptide or whatever. It doesn't matter what 9 amino acid peptide this helper T cell binds, because the superantigen is going to take this beautiful specific reaction and render it non-specific. And this is very dangerous because it'll cause vasodilation uncontrolled. And that's why we get the vasodilation during septic shock of all the blood vessels. That as the blood vessels get bigger, the blood volume stays the same. It leads to a loss of blood pressure that can lead to organ failure and death. So if we take a closer look at antibodies, we can see that they're Y-shaped, and there's a constant domain that macrophages have the ability to bind to. The constant domain of every antibody in the body is the same amino acid sequence. It's only the variable domain that binds the antigen. And that there are the domains that are different in every antibody, but the constant domain is the same. And this is like the part of the fork that the macrophage will grab onto to induce phagocytosis. And the antibody, another name for them are called immunoglobulin, and they're held together by disulfide bonds. We see the disulfide bonds here. There are these heavy peptide chains and light peptide chain. The part of the antibody that binds the antigens are called the Fab domain, Fab, and they bind. So this is IgG. It can bind two antigens at the same time. The constant domain is down here. This is what the macrophage, it's called the FC for constant, that the macrophage can grab onto that and phagocytose anything attached to the antibody. And again, I want to remind you that these antibodies are initially attached to the surface of a B cell, right? And then when they bind their antigen, they'll be inducing phagocytosis. And after the partner T cell interacts with the partner B cell, the plasma B cell will be produced, which will secrete 2,000 antibodies per second, and then the memory B cell and memory T cell will also be produced.